So early in the morning. Today we will present our diagnosis seminar, Spectronaut, the perfect partner for single-cell proteomics. My name is Monika and I'm a member of diagnosis support team, and today I have a pleasure to chair this seminar, and we have exciting program ahead. I will start with short introduction to our company and our products, and afterwards Chris Adams, Business Development Director for Bioinformatics at Bruker, will talk about integrated hands-free processing of single cell using combination of Selen1, EvoTIPS, timsTOF SCP, and Spectronaut 17. Our second guest speaker, Valdemaras Petrosius from the Cell Diversity Lab at DTU, will talk about enhancing single-cell analysis by tailoring DIA acquisition methods and using advanced data processing. So let me start with a short introduction. Our company has more than 14 years of the history in the innovation in the proteomics field, and it's mainly due to the fact that we have a strong R&D foundation. We are also proud to observe growing trust in scientific community, and this we monitor by growing number of the publications that are mentioning our products and technology. We have also one of the largest large-scale discovery proteomics facilities in the world, and this we are using for our contract research services. When it comes to our products, we can divide them into categories. We have kits, including sample preparation kit to enhance the reproducibility of your sample preparation, IRT peptide kit for the quality control, and PQ500 for the targeted quantification in plasma proteomics. Also, we have a separate group of our products, software, and here I will focus a bit more to tell you more details. Spectronaut is our flagship software for DIA proteomics. It features such workflows as library-based DIA analysis and direct DIA, including freshly introduced direct DIA Plus. SpectroDive is our solution for the targeted proteomics. It supports such workflows as PRM, MRM, and last year we introduced support also for the ion mobility technologies. And finally, SpectroMine is our software for DDA analysis, and it has integrated workflow for the samples labeled isobarically. While working on our software, we focus mainly on the three aspects. First, we would like to deliver the best performance for the software at the market. Secondly, we would like to have support and trust from scientific community, but this already I mentioned that there is a growing number of the publications, so we are happy to observe it. And finally, we would like to give you the best user experience to make your processing of the data easy and covering all the aspects you would like to have. And with this in mind, we were also working on the release of Spectronaut 17 that was released last December. It gives you the best performance in the library-free DIA analysis. It features a versatile generation of the libraries using Pulsar integrated search engine, and it has the broadest support for the PTM analysis. With SpectroDive, we achieved the best performance in the high-throughput targeted proteomics. It features machine learning assisted peak picking, automated FDR control, and IRT calibration assisted scheduling for the targeted proteomics. With SpectroMine, we achieved the best performance when it comes to the speed and identification rates. And as I mentioned before, it has streamlined workflow for the isobarically labeled samples, but also you can perform their library label-free quantification. And last but not least, I would like to show you our Quick software. Quick is meant for the quality control. It's a free-of-charge solution that works with the IRT peptide kit, and it allows you to monitor behavior your LC-MS in almost real time for your facility. For all our proteomics softwares, we offer 30-day trial licenses. They are assisted by our expert team, so if you are new to DIA proteomics, if you are new to our software, you will have a very good experience with the trial. We will help you to establish your workflow. And for the academic environments, we offer attractive discounts. as well as for the bounded purchase of the multiple softwares. And before handing microphone to our first guest speaker, I would also like to invite you to join us in the morning poster sessions. We have 2 posters featuring Spectronaut 17, and I will be presenting one, and my colleague Sira, and I also encourage you to come and give us tough questions, but also light ones if you prefer. And with this, I would like to hand the microphone to Chris. Chris, welcome.
Thank you to Biognosis for the invitation to speak. It's a pleasure to be here with you guys in the morning, bright and early. I heard there was a pizza crawl last night. Pizza usually involves pitchers of beer. Hopefully those were enjoyable as well. My former PI, he said the difference between academic science in the US versus Europe is exactly that, pizza and beer. So yeah, it's a pleasure to share with you guys some of the technologies that are coming together to dive deeper into the single cell. And I think you'll agree with me that over the past year or 2 or 3, some of that foundational work that existed has really allowed us to just go, leaps and bounds forward in such a short amount of time. So everything that kind of comes together is similar to this guy trying to crawl up this pegboard. Now, I don't know if you guys have ever tried this, probably not, but it's quite difficult. And that guy can do a lot of pull-ups, so that's not the problem. Strength isn't the problem. Sensitivity, the mass spectrometer isn't the problem. But it's all these things coming together, the pegs, the holes of the pegs, and then of course, just the strategy, software itself. So when all these things come together, you can actually get up that thing. And what I will show you guys today is how exactly to do that. So, with that, I want to start with the acknowledgments, right? Because I'm actually the fortunate one to be able to present this work that was largely done by a fantastic team, and so I'm really honored to be able to do that. Largely led by Christoph Crisp here. He's a Bruker team member, and he's been fantastic to drive all of this forward. So it starts with the launch of the timsTOF SCP. So this is a 1.5 year old, 2-year-old mass spectrometer at this point now. It's fundamentally different than the classic timsTOF instruments in the sense that, one, it has a larger bore, but more importantly, you'll see this high pressure ion transfer optics. So there's an entirely different pumping region that's happening. So that's many more ions can actually get into the detector in the end. And we've seen about a 4 to 5x boost in raw intensity. But as I said, strength in itself isn't enough. You need much more than just that. So one of those other advantages that we have is what we know as DIA passive, which has already really taken the field by storm, and we're quite happy with the performance here. There's a couple fundamental advantages that you get with the DIA passive. First and foremost, you get that additional dimension, right? So you can move the ions by both mass to charge and ion mobility, and that's really important for that selectivity. The second part is that you have passive, parallel accumulation, serial fragmentation, so that so that really boosts your duty cycle, so that when you're doing a typical single DIA scan, in our case you're doing 4x, right? So you can get many more scans across the MZ range with the specificity of the ion mobility selection as well. So we tested this with multiple gradients at very low loads, and I think that you guys will appreciate what we ended up finding was that at very low loads, gradient length doesn't give you a big advantage, right? So it trends to about a nanogram or 2, and then going out longer gives you a little bit of advantage, but at these very low loads, there's very little advantage going longer. So 15-minute gradients, 30-minute gradients, somewhere in this range is quite acceptable to get the full proteome advantages. And then coupled with the Selenium, the instrument from Selenium, they have this instrument that's been on the market for a while now, but it's been integrated into our platforms and others more recently. And this thing sorts cells by size, right? And it's very selective. And it can tell exactly where it wants to sort those cells. And that's really important for the next applications that I'm going to show you guys. Because it's actually, this is really the cool part. So Selenium worked with Bruker and then also EvoSep quite closely. And they came up with this, what they call Proteochip Evo 96. So the skunkworks name for this thing was calamari, because you see these little holes that stick out like an octopus's, whatever on the tentacles of an octopus. But the point being that these plates are on the dash of the cell one, sorting the single cells into them. Now, what's really important is to understand that there's actually two layers here. There's an oil layer on top, and then below that is all your digestion buffer, etc. So the single cells are moved into each well. Digestion, lysis digestion happens. Importantly is that oil layer. And what they figured out really creatively is that given the fact that the freezing temperature of oil is so much higher than water, then you can actually freeze these. And what happens is that the oil comes to the side, opens up a little pore, and then you have a way to get your digested single cell material out. So what I'm showing you here is a zoomed in section of a frozen plate where a single cell has been distributed into that cell. And then they then spin this, so they can take this plate when it's still frozen, which means 4 degrees C, put it on top of the Evo box, right? So where you have 96 plates, they line up exactly, do a quick spin, 10, 15 seconds max, and then that single cell material is distributed directly onto an Evo tip. And then this is the spun version where the digested single cell material is now escaped, but the oil still remains, right? So it's still frozen onto the sides. And it's really hard to see this, but that's exactly what's happening there. So this is really the technique that was being used. As I said, spun onto an Evo tip box of 96 well plates. So naturally, you use an EvoSep to do this from an LC side. For this work, it was the 40 sample per day. They have now extended to different methods, including an 80 sample per day, as well as you can go very slow at 20 samples per day. It's funny that I say 20 samples per day is slow. Five years ago, I would have just thought that's normal, but that's very slow now. So how does it look like when it comes all together? Let's go back to that guy climbing that wall, and then you'll appreciate that we now have the ability to really do single cell proteomics. So we, or the teams at large, went about this designing an experiment where they took advantage of the size of those cells that can be sorted by the Selenium instrument, right? So they sorted them, these are HeLa cells, and they sorted them in buckets of 18 to 20 microns, 21 to 23 microns, 24 to 26 microns, and then 27 to 30 microns. So even within the same cell population, there's quite a diversity in cell size. And that's exactly what you can see here on these different cells. And then this is an overlay of all of the LCMS runs that were performed. So they did this for 24 cells in each of those buckets. So a total of 96 cells that were run on the plate. Of course, all of this was searched by Spectronaut 17, and there's a lot of advantages using Spectronaut 17. One is the fact that you have the ability to use the libraries of your choice, and then the fact that you can do project-specific libraries. This is really important to us, and this is what I'm showing you guys here. So Bruker, we built quite a big library over the time, and this very large library that I'm showing you down here of more than almost 13,000 protein groups and almost 500,000 precursors. That's actually from highly fractionated, high reverse phase pH DDA runs generated from 2 cell lines, MOLT4 and K562. So different cell lines than HeLa that we're studying here. And then, of course, you can search all of your data against that library, which we did, and you can see the performance there. And then you can also create a project-specific library, which is what was done here, using 5 nanograms of HeLa. And in that, the number of protein groups is modest, about 5,400. And then the number of precursors is also quite modest. But we do increase in the number of proteins that we identify. And then, of course, you can then modify that library further by adding what is 8 single-cell samples that were run. Right? So altogether, this is what these different libraries look like. And then in the end, we actually then searched it with library-free or direct DIA plus in Spectronaut 17. And that's where we see very comparable numbers as to what we were seeing with the 5 nanograms of 5 nanograms, 8 single-cell samples that form. Of course, we searched this against a non-specific library, C. elegans in this case, and it was very reassuring to see that we see very little hits there. So across these 96 single cells that were processed according to size, the numbers are quite reproducible in terms of peptides, protein groups, as well as if you're very conservative, you can go back to the old days. I'm sorry, the print here isn't very good. This is proteins with requiring at least 2 peptides for observation, and even still, then we see more than 3,300 protein groups on average per single cell for each of those buckets. I also mentioned in the beginning the importance of that extra dimension, the collisional cross section, and both Spectronaut 17 does do the recalibration accordingly. So this is the retention time recalibration as well as the collisional cross section recalibration. You'll appreciate how linear this becomes, right? So it's really important to understand that collisional cross section is fundamental to these experiments. So the CVs are shockingly tight, considering that there's a lot of. I mean, these are there's 96 different biological experiments, right? So to think that your CVs are near or approaching 20% at the protein level is quite astounding. So we were also really happy to see that. And then now we start asking some more detailed questions. What does the difference between these cells look like according to their size? So this is a nice volcano plot that we put together. And you can see that if you look at these 2 smallest cell groups, there really isn't that many outliers that start to stand out. But then when you start comparing very small to the largest, the proteome starts really coming out to you to see all these different expression profile changes from a quant perspective. And it's interesting, I did some math last night and maybe the day before. And what I found, I actually didn't know this at all, but looking at some of these papers, it's assumed that per micrometer cell, that there's on average 2 to 3 e to the 6 proteins, right? So you can calculate the volume quite easily, of each of these cells, and you'll see even from the smallest to the largest cells, it's 3 times more protein that you could potentially see. So we were actually quite worried about this to ask what this means in terms of how to normalize, right? Because you can expect very different expression profiles accordingly. And we've done these experiments, both normalized and unnormalized, And I'm happy to say that actually, even in the unnormalized data, we still see very good distribution of those. So of course, we can also do a PCA analysis. And what we find here is that those two very small subset cell types, so this is the 18 to the 21, and then this is the 24 to 27, or I'm sorry, 18 to 21, 21 to 24 here. They have their own coordinate, and then whatever happens at that middle phase during cell growth and cell size really distributes them into their own quadrant. So that's really exciting to see. There's much more to do here in terms of following up on the biology, but that's not necessarily Bruker's responsibility. So we hope that you guys can take that to the next level and provide us with some critical insights at the single cell level. So I will again give thanks and recognition to the team who performed all of this work. It's been a fantastic collaboration, and I'll conclude by saying that single cell proteomics is still very difficult, but we're getting a lot better, right? Spectronaut 17 with Direct DIA Plus is a very powerful software tool to be able to perform all of these analyses. And then I hope I demonstrated that it really does take team science to get to the answer to all of these problems, right? So it wasn't Bruker, it wasn't just Bruker, it wasn't just EvoSep, it wasn't just Cell1 or just Spectronaut. It was all of these teams working together to get to the bottom of this. And then DIA Passive has massive advantages to digging deeper into the proteome by being enabled by that collisional cross-section, fourth dimension. Yep, and then let's find out more about the helibiology. So thank you. I think we're doing questions after. I'd be happy to take them.
Thank you, Chris. Indeed, we will have a joint Q&A session afterwards. So please hold your questions for later. I would like to invite our second speaker of today, Valdemaras, and he will talk about enhancing single-cell proteomics through his hard work in the tailoring data-independent acquisition.
Thank you. Good morning, everyone. Yes. So today I'm going to talk a little bit about what we have been working on in Irvin Schuf's Cell Diversity Lab in DTU, which is located in Denmark. Which the overall goal of the project was to tailor DIA methods to different amounts of limited input, and in the end use these methods for single-cell proteomics. So to begin with, why do we even care about trying to quantify the minuscule amount of protein encapsulated in a single cell? So if we really want to get a deep understanding of fundamental biological programs, we need that single cell resolution. If you look at the small cartoon example here that I have, if we have, for example, a tumor sample or just a tumor, it's composed of different types of cells, and all of them are unique in a specific way. We also have some spatial information. If we do population-based proteomic profiling, we get a rather aggregated response. And if we look at some stimuli from the tumor, we're also getting an aggregated response on the stimuli level. And although that insight is truly very valuable, I'm not saying that it's not. Single-cell resolution can provide a little bit more to that. So then if we are able to do that, we can actually study the profiles of all the unique heterogeneous cell types in the tumor and also study how they respond to whatever we want to do to the tumor. And although single cell, I think proteomics now is hitting a really exciting stage where we're finally moving further from just it being a technical development with showing numbers and then doing a little bit of a biology just for so it truly can be now leveraged for biological application. So it follows the same building stones as you would just have for bulk proteomics. So I have sample acquisition, preparation, data acquisition, and then analysis. And today I'm primarily going to talk about the data acquisition part and tether in a little bit also in the data analysis side. So you can pretty much split all the workflows for single-cell proteomics into two major categories. So they can be multiplexed or they can be label-free. So the multiplexed first approach that was developed by Budnik et al., so it was in Nikolai Stalov's lab, was a multiplexed approach where each single cell was labeled with a unique TMT, reagent and then combined, also with a booster channel containing a few hundred cells. And that was pretty much the genesis with a few extra papers of the single cell field, so it's 5 years old. More recently, the same lab also developed a Plex-D approach where you can use multiplexing with DIA by using the M-Q reagent. But my talk is primarily going to focus on the label-free side of things where you can either use DIA or DDA acquisition. There's like a few references that so far have reached the biggest proteome depth with these methods. So just a refresher for those who missed the LFQ Battle Royale that was on the first day. You can label-free, you can do either with DDA or DIA, and in DDA you just select the most intense precursors for further fragmentation and you generate rather these clean spectra, although they are chimeric to some extent. While in DIA you sequentially isolate all different segments of MZ and in theory acquire the spectra of all the precursors present in the sample. And I think it's well accepted that you start increasing that isolation window more and more. What you're doing is you're co-isolating more and more ions, and that's getting hairier and hairier and more difficult to interpret, and that in the end really detrimentally affects your protein IDs. But we kind of hypothesize that although these co-isolation constraints are quite bad when you have high loads, if you're working below 10 nanogram, or large isolation windows might actually be preferable to small ones. And that was pretty much what I aimed to test in this project. So to quickly test that, I generated a panel of DIA methods that have different isolation window sizes. So the MS1 resolution is kept constant at 120K. And then I varied the MS2 resolution to 50, 30, 60, 120K. And then increased the also isolation window, keeping the cycle time of the scan exactly the same. And if we look at different loads, so we have 100 nanograms, 10 nanograms, and 1 nanograms. If we do the 100 nanograms first, we see that around 30K and 20 MZ isolation window, we're hitting our optimal value of around 4,000 protein groups. And if we go further, we see that our protein coverage is decreasing due to the isolation windows being too large. However, if we dilute our input 10 times to 10 nanograms, we can really nicely see that we're shifting the optimal isolation window to 40 MZ and the resolution being 60K. And we see the exactly same effect once we do a 1 nanogram input with the optimal method being at a really large 80 MZ isolation window and 120K resolution. And although the co-isolation effects, they didn't disappear or go anywhere, the benefits you get from the extra resolution and injection time just simply seem to outweigh them. So if you have low input, what you want to do, you can really use these high isolation windows to get the maximum protein coverage. So to push this a little bit further, I adopt a specific flavor of DIA, which is known as high resolution MS1 DIA. So what you kind of in normal DIA, what you do a survey scan, so an MS1, and then you sequentially isolate all your MS2s. But in HRM MS1, what you do is you insert a few additional MS1 scans into your acquisition method, and you sacrifice any ability to do MS2-based quant, and you use only the MS2s for that identification. And that changes your cycle time because you only have to then calculate it between the MS1 scans, but you have 3 times the time to actually spend on your MS2s. So you can either have 3 times narrower isolation windows, or you can have 3 times longer injection time. And if we just compare how the two methods perform for 1 nanogram, if you look at the peptide level, with standard DIA, we're getting around 5,000. If we use HRM-MS1, we're getting close to 9,000. And on the protein level, we're also pretty much doubling our protein coverage. And if we look at the precision of the methods, so this is MS2-based quant on the standard DIA method, MS1-based quant on the OT, we can see that the vast majority of our C values are way below 10%. So it has really good precision in itself. But to give standard DIA also a better chance, I repeated the window isolation survey also with standard DIA and HRM-MS1 to see what were the optimal values. And I pretty much observed the same thing, that on 1 nanogram, the optimal method seems to be around 120K. We're getting 1,500 protein groups. However, with the HRM-MS1, we can kind of boost that further close to 2,000 from 1 nanogram. And because we also have a much narrower isolation window, we're maintaining the same cycle time. For standard DIA, we have to use 120 MZ. With HRM-MS1, we can get away with using 40, and that boosts our protein coverage. Yeah, and then at that point, we kind of got hit by the perfect storm. So Thermo Fisher released micropacnium low load column, which it's absolutely amazing, and no, Thermo is not paying me to say that. So far, I started using this column approximately June 2022. I have more than 2,000 samples on it, and it's still going strong, and so there's no sign of dying anytime soon, so it's absolutely amazing. And then also Spectronaut 17 came out around the same time. So if we compare, so we made two methods on these columns, so a 32-minute and 52-minute method, so that corresponds to approximately 40 and 24 samples per day. We can see that we're getting really nice coverage from 20 nanograms. We can get close to 5,000 protein groups and going down to almost like somewhere you would be in the level of single cells of 250 picograms. We can reach around 1,700 protein groups. But then if we research the same data with Spectronaut 17, we saw a really nice boost in our IDs. So suddenly we gained 1,000 proteins on the highest load of 10 nanogram, and we got an extra close to 500 proteins on the lowest scale, showing that Spectronaut 17 indeed can really nicely boost your proteome coverage. So one extra thing you can do to drastically increase your IDs for low input sample is to use a library that has been used from a higher load sample. So, but we're kind of a little bit skeptical. What is the quality of these extra IDs you're gaining if you're using a much higher load library on your low load? So we made a simple Silek experiment where we just grew HeLa cells in light or heavy media, made some peptides and made 3 mixes. So 1-to-1 mix, 1-to-2 mix, and a 1-to-5 mix. And then combined the heavy and light and told it to yield a 10 nanogram input sample. And that we consider our high load sample, so the 10 nanogram. Then we diluted 10 times and made 1 nanogram samples, and that's our direct injection. So the run samples and also to generate a gas phase fractionated library. And you might be wondering, what is a gas phase fractionated library? So this is an approach that has been developed by Brian Searle, and I think it's a really elegant way to do a fractionation on samples we're not allowed or you don't have the capacity to do a fractionation. So instead of looking at your whole MZ of range of interest in one run, you split it into 6. So you, for example, if you're interested in 600, you do 6 injections only looking at 100 MZ, and you just rerun the same sample 6 times. And that means that you can actually spend more time, so you can spend 6 times the time on your MS2s, MS1, have narrower isolation windows, you have more time to actually do the data acquisition. And so you can have more proteome depth with constructing a library like this. But so, onto the actual data that came out from this. So if we did the direct search, we got around, from 1 nanogram, we got around 1,000 protein groups for this. And if we look at the quant accuracy, it seems to agree more or less. It seems to be going down a little bit if we're really drastically decreasing the amount of light peptide we have in the sample. And if we actually add a 10 nanogram library into the search, we can see that we're pretty much doubling our IDs. We're getting like 2,000 something. It looks pretty great. But if you look at the quality, these peaks suddenly they become really, really flat. And that means that overall the quality of the quant quality of the whole data set was pretty much cut in half. Which is not something that you kind of want to see when you're doing biological inference. So the question was, okay, so what's the quality of purely the extra IDs that we're getting? So in the top here, you still see just the IDs that we get by direct DIA. And this is just the same distributions, but all the proteins that we can find by direct DIA without the library are subtracted. And you can see that overall, the quality of this data is not very good and should be interpreted with some caution. It's not absolutely always wrong, but you can see that some of the proteins are actually, the peaks are still in the middle, but overall the distribution are really flat, showing that you have a high probability of getting the quant value wrong. Yeah, so with those things kind of established what we can, what we can and we cannot use, it kind of shows also to go with the gas phase fractionated libraries to maybe boost our numbers a little bit, because it's also something that you can do with single cell. And we prepped all the single cells with really standard lab equipment. So if you have a fax and if you have an auto dispenser pipette, you can, on a 384 well plate, you can replicate our whole protocol in the sample. It's quite straightforward. You don't need a cell-in-one or any fancy adapters or anything like that. So we just sort the HEC293 cells in a 384 low bind plate. We add with a pipette digestion reagents, and we just straightly inject with a U3000 the micropack column into the instrument. And we're running at a gradient of 40 cells per day, more or less. And if we look at the direct results, so this is a data set of 100 cells, just as a proof, we're getting close to 1,700 protein groups with direct DIA per single cell. If we supplement them with the gas phase fractionated library, so for single cell, the gas phase fractionated library is made in a way that we just run one single cell and then look at 100 MZ. So we kind of run 18 cells to have like a triplicate for all the MZ range, and then we just supplement that data into the search. So then we want to gauge, okay, so are we actually capturing any biological variation in this data set? So we use two methods to kind of cluster it. So we use the parametric method, PCA, non-parametric UMAP, the most popular approaches you see. And then for my PhD, I really like this one protein called MKI67, which is a G2M marker. So it's really highly expressed in late G2M cells. And if we look at the PCA1, we can kind of see that the color gradient really nicely follows the MK167 expression. And we're capturing a really large degree of variation in the data as well in that principal component, like 63%. And that makes a lot of sense because the only major biological variation that we have in the system is the cell cycle. If we look at the UMAP, we see a similar picture. So we have a kind of cluster that's slow in MKI67, so the G1 cell population. And then we have like increasing another cluster that's increasing in MKI67, which is probably cells going through S phase and more into G2. Yep, so with this, I would like to sum up. So for conclusion is you should definitely design your DIA method based on how much you're injecting into the instrument. And that precursor level quant works really, really great if you have low amount of input. And that gas-phase fractionated libraries can be used as a viable alternative when you cannot really fractionate your sample, and that the whole workflow is based on really standardized lab equipment, that you don't have to buy mass spec instrument that's specific for single cell. You don't need a specific single cell sorter for it. And finally, that Spectronaut 17 can really help you maximize what you're getting out of your data. I had a poster yesterday, so I'm not there anymore. Please ignore this. But I have a preprint up, and if you want more details on the quality and everything else that has been done that I haven't shown here, please feel free to look at this. And with that, I would like to thank my supervisor, Evan Schoof, and everybody else in the Cell Diversity Lab, the Porsche Lab, which helps us a lot with the biology side of our single cell efforts, the DTU Proteomics Core, Mo Fisher for making a really great column, and of course, Biognosis and Tejas Gandhi, which is extraordinary on point with any issue I have with Spectronaut 17, and of course, all our funding bodies, and that's Erwin, that's me, and Pedro and Nil are our gurus at Sample Prep. And with that, I would like to thank you for your attention.
Thank you very much for the fantastic talk. And before we start to open our Q&A session, I would like to invite you to go to our website whenever you have questions about our presence at this conference or questions about any of our products. And now I would like to invite both our speakers over here so we can start Q&A.